Hello everyone, a very warm welcome back. Today we are going to discuss anatomy in case of pteridophytes. First of all, let me tell you what the literal meaning of anatomy is. Anatomy is derived from two words ana and tome. Ana means up and tome means cut. That means if you are studying something after cutting it up, you are studying it as anatomy. Simply stated, if you are studying the internal structural details of animal tissues or plant tissues, you are studying their anatomy. Okay. The vascular cryptogams, I mean pteridophytes, they possess vascular tissues. Since they possess vascular tissues, it is right to call them vascular cryptogams. Okay. Cryptogams means those plants in which the sex organs are hidden, in which flowers are not produced, in which seeds are not produced. Since the pteridophytes, they do not form the flowers, they do not form the seeds, but on the other hand, they contain vascular tissues. It is not wrong to call them vascular cryptogams. They are aptly named as vascular cryptogams. You may be asked in the examination, vascular cryptogams are, and the options will be like this: pteridophytes, bryophytes, algae. You have to remember that the vascular cryptogams are the pteridophytes because they possess vascular tissues and they are cryptogams. That means their reproductive organs are hidden. Okay. And the vascular tissue in case of these pteridophytes, it is composed of xylem and phloem as usual. However, you have to remember that in these plants the cambium is absent, and this is the reason that secondary growth in case of pteridophytes is impossible. Okay, because it is cambium, it is this meristematic tissue which helps us in the secondary growth of stems and roots. Okay. Now, first of all, the composition of xylem in pteridophytes. The xylem in case of pteridophytes, it is composed chiefly of tracheids, but there are some exceptions. For example, Selaginella, Equisetum, Pteridium, you have also vessels in the xylem. When we will study our next unit, you will come to know the differences between tracheids and vessels. Okay, and for for this moment, you have to remember that in case of pteridophytes, the vessels are absent, but there are some exceptions. For example, Selaginella, Equisetum, and Pteridium, in which these vessels are present. Okay. Now, the composition of phloem. In case of pteridophytes, the phloem is composed of sieve cells only. Sieve cells are less specialized cells. These are present in lower plants, but in case of higher plants, for example, angiosperms, you do not have sieve cells, rather you have sieve tube elements. And these sieve tube elements in angiosperms, they are joined end to end and they form sieve tubes. And these sieve tubes are completely absent in case of pteridophytes, but there are some exceptions. For example, you are having Equisetum orvens and Equisetum giganteum, in which sieve tubes have been reported in the phloem. Okay, but in majority of the cases, you have to remember that only sieve cells are present. And one more important feature of the phloem of pteridophytes is the companion cells are altogether absent. What are companion cells? Companion cells are the cells which are in close contiguity, which are in close connection with the vessels of higher plants. And when we will study the anatomy of higher plants, you will come to know the structure and the function of these companion cells. And for this time, you have to remember that in pteridophytes, the companion cells are altogether absent. First of all, we will discuss the anatomy of roots. The roots of pteridophytes, they possess stele. Stele means the vascular cylinder. Okay, and the stele is composed of the xylem, phloem, and the pericycle tissues. Okay, stele is not only constituted of xylem and phloem; it also contains the pericycle, which surrounds the xylem and phloem. They possess a stele which is bound by endodermis and pericycle, and the xylem in case of these pteridophytes, it is exarch. Let me tell you what exarch means. If you are having xylem and that xylem shows the direction of maturation is towards inside, that means first of all the xylem elements which are directed towards the periphery, they become mature, and then the inner elements, the inner xylem cells, they become mature. This is known as XR condition. Okay, this is also known as centripetal development. That means xylem matures first towards the periphery, and then the inner elements towards the center, they mature later. Okay, this is known as XR condition, and the complete reverse of it is AND-OR condition, in which the direction of development is towards outer side. That means xylem elements mature first in the interior, and the peripheral elements mature later. Okay. Now one more important point about the root anatomy of pteridophytes is many ferns, they show di-R condition. They show di-R condition of xylem. It means the xylem, you are having two distinctive protoxylem groups here, one and second. Okay, you are having two distinctive protoxylem groups here. I will tell you, for example, this is one protoxylem group and this is second protoxylem group, and here in the middle you are having metaxylem. If you are having two distinct protoxylem groups in the root, this condition is known as di-R condition, for example here. Okay, and in some cases, for example equisetum, you are having tetra-R condition. You are having four distinctive protoxylem groups, and this is known as tetra-R condition, example equisetum. Now the stem vasculature, stem anatomy. The vasculature of stems of different pteridophyte species, it varies a lot, and accordingly you are having different types of steels in different species. For example, the first one, which is the simplest one, it is the protostele. In protostele you are having a solid core of xylem, and this solid core of xylem is surrounded by a ring of phloem. Examples are Selaginella and Lycopodium. The second type is siphonostele. In siphonostele, you are having a central core of pith, and this pith is surrounded by, for example here I will tell you, this is pith and this is surrounded by a ring of xylem, and this is surrounded by a ring of phloem. This is known as siphonostele. Here you can see, this is pith, this is xylem, and this is phloem. Okay, this is known as siphonostele. Examples are equisetum and ophioglosum. Okay beta, we are studying the anatomy of stem in case of pteridophytes, and in some cases you are having solonostele. Solonostele is the type of siphonostele in which you are having widely spaced, widespread leaf gaps. Okay, that type of siphonostele in which the leaf gaps are widely placed, they are widely spaced. Okay, they are scattered. For example, here you are having leaf trace and you are also having leaf gap, and these leaf gaps are widely spaced. Okay, this is known as solonostele, and the example of solonostele is in the rhizome of Marsilia. That means the underground stem of Marsilia shows this condition of stele, which is known as solonostele. Okay, solonostele is actually a type of siphonostele. That means you are having this central core of pith, and that central core of pith is surrounded by xylem and phloem. But in addition to this, there are widely spaced leaf gaps. Okay, then one more type is dictyostele. Dictyostele is the type of siphonostele in which you are having a series of interconnected, you are having a series of strands, and the individual strand is known as meristele. Okay, that type of siphonostele, that means you are having, first of all you are having the pith, and then you are having a series of vascular strands, and in each strand you are having both xylem and phloem. For example, this is one strand. Okay, this is the xylem, inner xylem, and this is outer phloem. Okay, that means each of these strands, which are composed of both xylem and phloem, they are known as meristeles. Okay, and the examples are Dryopteris and Pteridium. And one more important point that you have to remember about the dictyostele is, in case of dictyosteles, the leaf gaps, they are overlapping. ठीक है, leaf gaps are overlapping in case of dictyostele, but in case of solenostele the leaf gaps are widely spaced. Now leaves. The leaves in case of pteridophytes, they receive vascular supply from the main stem in the form of a leaf trace. That means here you can see this is the leaf trace. That means the vascular supply that arises from the main stem towards the leaf. The tissue, the xylem and phloem tissue that goes into the leaf from the main stem, that is known as leaf trace. Okay, and the gap which is left when a leaf trace is formed, when a leaf trace is supplied to the leaf, that is known as leaf gap. Here you can see this is the gap and this gap is known as leaf gap. Okay, and these leaf gaps are present in some species and in other species these are absent. You have to remember that the vascular supply that is given to a leaf from the main stem, that is known as leaf trace. Here this is a leaf trace, and the gaps which are left in the vascular cylinder of the stem, they are known as leaf gaps, and these leaf gaps are present in some cases. For example, here the leaf gaps are present and here the leaf gaps are absent. Okay, now the anatomy of leaves. The leaves in case of pteridophytes, they are composed of chloroenchymatous mesophyll. Chloroenchyma is a type of parenchymatous tissue. Okay, and that parenchymatous tissue which is present in the leaves, that is known as mesophyll. Okay, chloroenchymatous mesophyll. That means those cells which are green colored because of chloroplasts. Okay, and this chloroenchymatous mesophyll, it varies. In some cases, for example, in aquatic species, you have a homogeneous chloroenchyma. That means the tissues of leaf, the cells of leaf, they are homogeneous throughout. There is no distinction. There are no differences between the upper and the lower region. Okay, the upper and lower region is having the same type of cells. Okay, and this happens in case of aquatic species. And the second type is, in this type, the mesophyll, the chloroenchymatous mesophyll, it is differentiated into a spongy and a palisade parenchyma. The spongy parenchyma lies here towards the lower epidermis, and the palisade parenchyma, it lies towards the upper epidermis. ठीक है बेटा, palisade parenchyma में क्या होता है बेटा? Palisade parenchyma में you are having elongated cells. You are having elongated cells, and these cells are directed towards the upper epidermis, and the spongy parenchyma, it is located towards the lower epidermis, and this spongy parenchyma, it is having a lot of intercellular spaces between them. The cells of the spongy parenchyma, they are known as spongy parenchyma because a lot of intercellular spaces are present between the cells. But in case of palisade parenchyma, the cells are closely placed to each other, they are closely packed, and the individual cells are elongated. लेकिन spongy parenchyma में क्या होता है? Cells are circular in outline.